Mm. Hello, we'll be talking about biochemistry. So I discussed in the last course, which is different morphology. I told you virus are these strands of DNA or RNA that actually have some mechanism that is protecting them. This can come in form of the envelope, the spike. All these things are part of the biochemistry of virus because they are the ones that help the virus to navigate the human or animals or plants or bacterial systems that they are affecting. So once they enter, how do they get in? So now I'm going to discuss on how they enter, reproduce themselves, and do everything they do in their host cell. Now what happens is the first thing that happens is entry into the host themselves. They are not the one that enter the host, they are just about that somehow in nature. So when you consume them, you've consumed them, or when they come through the air or they come through, because they don't have legs, they are lifeless, like they are lifeless again. So they just enter the body. So once they now enter the body, they now start to replicate themselves again, because they are like the smallest part, like they just keep replicating themselves. They are not conscious about it, unlike every other cells that can be conscious against one stimulus or the other. They just have instructions and stuff designed them to protect themselves, to protect them against anything. You get what I'm saying? That could cause them not to. So the first thing that happens is entry, and entry I have to from entry into the host and entry into the host cells. Now I'll talk about entry into the host, which is when the proton, whatever born, that's when it's transmitted to the human being. It is transmitted to, then it now gains another level of entry into the cell. That's entry 2. If it has not gained entry 2 and it's flushed out, it doesn't have any effect. It's going to be outside. So in entry 2, to gain entry 2, it has to fight a lot of things. First, it has to evade the immune system. 2, it has to evade any digestive stress or whatever. This is why envelope and nucleocapsid are very important. So but once it has successfully been able to escape, it will move all around until it sees a cell that has its actually the cell that is targeting that has its entry port, and the entry port is like the disguise, the camouflage that this virus can use. Like I can camouflage into this one as its food because I look like it's food, or I can camouflage into this one as it's this because I look as it this. That is the major thing. So the body determines where it goes to. You get the entry port determines where it goes through, but any cell can be affected with any virus, in as much the cell has in all the requirements of life. You get what I'm saying now? So once it now gains entry, there are different forms of entry. There's the whatever mediated and whatever endocytosis or whatever some complex names like that, like receptor mediated endocytosis and some other things under biology. So although it's not just under biology, there's kynocytosis, there's kinesis, there's different types of at which it enters into the bacteria. Then once it enters into the bacteria, let's say the first entry, which is into the host cell, and I've explained the immune defense and camouflage part, then the entry into the cytoplasm of the cell. So once it gains entry with everything that remains. You get everything that remains from the attack of or whatever that remains from the attack of antibodies and whatever antigens and the attacks of or if it's successfully camouflaged, whatever enters into the cells with the remains, which is mostly the nucleocapsid. Now the cytoplasm start another attack on it. You get, because one, don't forget, every cell has its own innate immune system, and the immune system is not naturally targeted at immune system. Sometimes, most of the time, they are targeted at food. So it may go into the Golgi apparatus, go into whatever. So it can gain entrance into 2 types of nucleus. It can either gain into the mitochondrial nucleus, which is, or into the cell major plant nucleus. You get what I'm saying now? So when it gains entry, then what happens is that when it gets entry into the this thing, then it degrades. Let's say you get, once it enters the nucleus membrane, then degradation happens. If it goes there with the nucleocapsid, if it doesn't, then it means it has been degraded on the way. But once it enters there, the chemical component of the nucleocapsid is reversed in nature to that of the cytoplasm. That keeps it safe till it gets into the nucleus. So once it gets into the nucleus, it reverses back. You get what I'm saying? And once it reverses back, in fact, it can create a reverse RNA, whatever. So once it actually reverses back, it degrades and disintegrates, and then leaving the DNA of the organism in the nucleus. Now that is where it is. It has entered. The entry has completed. The entry is not aimed at the cell. The completion of entry is the nucleus itself. It enters the body, that's entry 1. It escapes immune system, whatever during the entry 1. It enters the cells in entry 2. It escapes immune system in entry 2 before it now finally enters the nucleus. And when it enters the nucleus, it doesn't need immune system again, because it has entered the palace. It doesn't need to compete with the DNA, because the DNA is just a strand, like a strand containing a programming language. So a new program has entered. Now it will now start using the normal DNA replication process that we all know in genetics to replicate itself. You get the replication, transcription, whatever, all those processes in genetics, in DNA replication or RNA reverse replication. Like, that is how it happens. Then when it happens that way, then it goes down. It keeps reproducing itself, like keeps carrying out replication inside the nucleus, and at the same time keep producing outside of the nucleus. It keep producing toxins that are strong enough to weaken the cells, the external membrane, the cell membrane of the cell. While both of them are happening like that, it's continuous like that, little by little, until it gets to the point where the cell of the nucleus has become so full that it has already put more pressure on the cytoplasm of the cell outside, and then it's ready to burst. Two things happen. If both membrane have the same strength, or close to the same strength, then resonance breakage happen. Both of them break at the same time. That is explosion, and the cell is liberated. If not, the external one is liberated, meaning it will now remain the cell nucleus floating around at that location. So once that happens, it is definitely going to float around, because it has no more attachment. During that process, antibodies may actually launch an attack on the membrane in order to degrade whatever is causing